Please give Dr. Yoon a warm welcome.
Thank you very much. It's always a pleasure coming to these talks because as a clinician, I know very well how important families are to the well-being of our patients. So I really enjoy giving these talks periodically. So, in addition to talking specifically about the two research projects, I hope to contextualize these experiments or these studies and give you a broad review or sort of update of some of the more recent findings in the field, as well as highlight the ongoing challenges, the difficulties we face in trying to identify causal mechanisms. And I hope I'll have some time at the end for questions. So, I won't have to belabor this. We all know how impactful this is, but I think, especially given that we live in this country, putting things in dollar terms is certainly attention-getting. And so this is an incredibly high number. So one of the main reasons why it costs so much, as we all know, is that we don't have great treatments. They are partially effective for some patients, for some symptoms. Other reasons include the fact that we don't have objective or quantitative tests for diagnosis or monitoring of illness state. We don't have treatments for the most disabling symptoms, such as negative symptoms or cognitive deficits. And then no truly novel treatments have come to the market for decades now. And this latter issue has been fairly well discussed in the literature, including by Dr. Insel in a number of publications. So one of the ideas that have gained a lot of currency in recent years is to develop what we call mechanisms-based treatments. And so this slide outlines this idea that once we discover mechanisms, we can target new treatments related to the mechanisms. And then we also may develop new mechanism-based diagnostic tools. However, if it were that simple, we probably would have done it right now. I think a number of challenges exist, and I'll talk about just a couple of them right now. One of them is this. If you think about, in the case of schizophrenia, or really most psychiatric conditions, the core problems involve higher order, likely human-specific abnormalities or domains of mental processes. And so what is a delusion? It involves beliefs, convictions. What is a hallucination? It is a perception of a false sensory or environmental stimulus. And you have to be able to report that perception. And so simply put, a mouse, it's impossible or nearly impossible to model and study these phenomena in animals. And in the rest of medicine, for example, in cardiology, in nephrology, where the core deficits and symptoms are in other domains, let's say blood pressure, temperature, where you can measure things and therefore measure these properties in animals, that task of finding causal mechanisms is much, much simpler than in the case for psychiatric conditions. So what's the alternative? The alternative is to study these processes directly in humans. However, that's also problematic. And this figure illustrates this, and let me just go through this a bit. So this represents sort of the resolution of sort of the elemental unit in a neuroimaging scan. This is a voxel. This is a 3D version of a pixel, something that you'd be more familiar with. So within this voxel are all these different types of tissue, neurons, and cellular processes that may be occurring within this tissue and between these cells. And so I think this illustrates the idea that with human studies, with using non-invasive methods, we simply lack the resolution to detect pertinent processes that are occurring in the brain. So what's the solution to this? And this represents, one way forward out of this conundrum. And this is termed by some as called reverse translation. Well, translational research usually means translating findings from animals into humans. For example, you screen a bunch of molecules in rodents or other animals, test it for efficacy, see if there's target engagement, modifying the process of interest, and then translate that into humans. Again, for the reasons we just discussed, that's difficult to do in psychiatric conditions. Reverse translation is sort of the reverse, where we first identify key elements or processes of interest, and then reverse translate or hand that off into the area of animal studies, such that you can get down to some of the really nitty-gritty detail, specific neural processes that may be involved. And so globally, or in a large frame of reference, that's the type of work that I and a lot of other of my colleagues are attempting to do to deal with this issue. Is that first define in a literally and figuratively a larger scale in humans what the pertinent processes are, which may then serve as a clue to animal researchers who can then further define the neural basis of the illness process. So let me talk to you about two areas and two specific experiments that I'm currently conducting at Stanford. The first involves GABA. GABA is the major inhibitory neurotransmitter in schizophrenia. And there has been one of the most prominent hypotheses or theories about the neural basis of schizophrenia is termed the GABA hypothesis. And in simple terms, this hypothesis proposes that there's a deficit in GABA function, synthesis or function, that results in brain dysfunction that gives rise to some of the symptoms of schizophrenia. Most commonly, we think that GABA is related to cognitive deficits, but it's also possible they may be responsible for some of the other symptom domains in schizophrenia. We don't really think about it that way because, firstly, all these systems are all intertwined and not necessarily in sort of like a linear or simple opposing ways. So there may be connections, but it's probably going to be very, very indirect. So let me give you just a brief background on some of the key findings that have informed the basis of the GABA deficit hypothesis. So if you could focus your attention on here, this is really the main part of the slide I wanted to focus on. This is a figure from a post-mortem study. A human study, but post-mortem means that we take sections of the human brain and study it. I did forget to mention that one of the caveats about the limitation of human studies is that you do have the option of or the possibility of conducting post-mortem studies where you can get at sort of more sort of fine-grained neurobiological detail. However, there's obvious limitations to that approach. So this is a post-mortem study in which they stain for a key enzyme in the control and metabolism of GABA function. What's called GAD67 is one of the major synthetic enzymes. So if there's not enough GAD67 or this enzyme, you don't have enough GABA produced in your brain. So they stain for this enzyme here in a healthy individual, and these light areas, or like stars, are areas that have GAD67, or the mRNA for GAD67. This is an example of an individual with schizophrenia treated in the same way, who chose a significant lower density of GAD67, approximately 30%. And so from this, we infer that there's a deficit in GABA production and deficit in GABA function. So then why is GABA so important? So this figure will illustrate this. So first, these reddish pink lines represent the action potential or activity. So one of the major ways in which the neurons function or promote its activity is to activate and send an electrical signal that can be measured using tiny electrodes inserted into the cells. And what occurs, or these spikes, are tracings of the action potential of one neuron. And as you can see, there are many lines, meaning that the action potentials of many neurons are being recorded simultaneously. So if you look here, see that each individual action potential is not lined up. It's sort of cacophonous. And the way I sometimes think about it is sort of like an orchestra. If you ever go see an orchestra or any band or group ensemble, before the performance, before the conductor comes on stage, they're tuning or playing their own thing, and it creates a cacophony. Now, that is in contrast to what happens here. These black lines represent firing or activation of GABAergic neurons, so neurons that use GABA, and these are these black cells down here. And so one way to think about it is that they're pace setting these neurons. So these tiny GABAergic neurons synapse across multiple cells. So a single cell has the capacity to pace set the rhythm for many, many different cells. So the idea is that these GABAergic cells, the reason they're so important is that it helps organize into a synchronous, organized rhythm a large number of what's called pyramidal cells, sort of like a conductor, saying each neuron has to fire simultaneously. Yeah, I'll show you that a little. Yeah. So that's the driving idea, that this is a fundamental process in the human brain that allows us to do a number of different things in an organized, coherent manner. Perceive, talk, walk, move in an organized manner. And so with diminished GABA function, the thought is that there's more of this. And actually, I've drawn that out to make it very explicit. So in the upper case, it's very similar to what I showed in the prior slide with intact GABA functioning, and you have sort of this organized wave-like activity. And here, the smaller arrows, black arrows, are meant to represent diminished GABA functioning, therefore disorganized activity. So one possible example of this dysfunction is anti-NMDA receptor encephalitis. How many of you are familiar with this entity? Is that? Yes. So we're not 100% sure exactly how this occurs, but one of the main the leading theories is that the cells or the neurobiological basis of this phenomena is dysfunction of GABAergic cells. Because while NMDA receptors, it's a major class of neuronal receptors, appear everywhere in all different cell types, the key cell type that is thought to be involved is in GABAergic cells. So when you have dysfunction of these neurons, it disables the GABAergic cells, which then leads to disorganized neuronal firing and sort of the panoply of bizarre, unusual, disorganized behavior that is often seen in this condition, and as well as in other psychotic disorders. So, we've contributed to this literature or this theory. Before I showed you an example of a post-mortem study. There are obvious disadvantages or limitations, as I mentioned. One of which simply could be that any finding you find post-mortem may be having something to do with the dying process or proximal process related to death. May not be specific to the illness itself. And so we did an in vivo study, because in this case using in vivo neuroimaging, and there's a way to measure GABA in the living human brain. In this study, which we published about 9 years ago, we sampled the back of the brain, in this case the visual cortex, shown here, and then compared the amount of GABA in that region in healthy individuals versus individuals with schizophrenia, shown here. So healthy is dark, people with schizophrenia, gray, and showed a fairly robust difference. And this is sort of for those of you who may be interested, this is a spectra. It's sort of the chemical sort of signature of GABA shown here. Yeah. Hello? Okay, I think I turned it off accidentally. That's exactly right. We could talk about it a little more if we have time later, but I think the deficits that exist in schizophrenia are mostly pancortical. They may be most prominent and most important in other areas, but they seem, at least in the case of GABA, they seem to be replicated in other areas of the brain. So the prior study I showed you was in individuals with established chronic illness. So then the question becomes, well, this related to illness chronicity rather than the illness itself. And so to address that potential confound, we recently completed a study involving recent onset individuals with schizophrenia within 1 year of illness onset, using very similar methods, sampling the visual cortex. So we replicated that deficit. In this case, you may notice the effect size or the magnitude of the difference is not quite as large for chronic. So that leaves open the possibility that there's something with the illness process that may be further decreasing the amount of GABA and presumably GABA functioning in schizophrenia. No, it's all non-invasive, in vivo. Yes. I'll try to talk about, no one really knows in the end. There's some ideas, but I'll talk about that in a little while. So, the relevance of these types of findings is that it's ultimately in terms of treatment. So this leaves open the possibility that one potential treatment, a novel treatment, would be through modifying, improving the GABA system. Excuse me? Oh, parts per million. Yeah. Yes. So I am sort of there are a number of limitations or challenges, but one of the most significant, however, which I'm sort of glossing over, is the fact that these non-invasive in vivo methods for measuring GABA, it's noisy. It can be difficult to pick out the signal. And I won't belabor or go into details, but to show you this graph. So I showed you the nice group average results before, but here's an individual subject. And hopefully some of you will notice that this is sort of the GABA peak. And you can see clearly that it's very noisy. You get sort of the other areas, right? You can barely perceive this above sort of background signal. And so another investigator did a recent meta-analysis where they collect, all the studies that have been conducted in a particular field, and they look to see if they're at a group level, at this meta-analytic level, whether there's consistency and determine the robustness of the findings. And unfortunately, at least in the prefrontal cortex, most of the studies have been done in the prefrontal cortex, by the way. I and a few other colleagues around the world have been the sort of the outliers. At least in the prefrontal cortex, the combined effect size is very small. Still in the direction of diminished GABA levels in schizophrenia, but it was approximately negative 0.3. For those who are familiar with statistics, that's considered pretty modest or small difference. And so this presents a really significant problem for a number of reasons. And so this motivated this new study that I'm undertaking at Stanford. All the prior studies that I showed you before were done in resting state, meaning the subject lies in the scanner, hopefully not falling asleep, but not engaged in any task. So in a resting state. And that's typically 99.9% of all spectroscopy studies like this are conducted that way. But recently it's become apparent that the enzyme or the levels of these metabolites, including GABA, glutamate, and a host of other neurochemicals in the brain, are actually dynamic, meaning that their levels rise when that a metabolite is used, and that we could measure it. So when I found out about this, I immediately thought that this could be applied. This idea of measuring task-evoked or dynamic GABA could be applied or tested in schizophrenia. Because as I mentioned to you earlier, one of the core findings in this area has been decreased GAD67, which is an enzyme that produces GABA. So my idea was that if that's one of the core findings, maybe a related deficit is that the inability to mobilize and synthesize new GABA when the brain is taxed or has to produce new GABA. I'll talk about that in a little while. It's all NMR or MRI-based scans. So the hope was that maybe this would produce a more robust differentiation and reveal the underlying GABAergic deficit in schizophrenia. So I included this figure just to give you an illustration of the task or what we're having the subjects do while they're lying in the scanner to sort of evoke or have the brain work in order to produce more GABA. And as I mentioned to you earlier, through its function in organizing activity, I think you could probably ask them to do a number of different things in order to engage GABA cells and have more GABA being used. In our case, as this one audience member mentioned, we were interested in the prefrontal cortex GABA, because the reason being that we think that GABA functioning in that region is so important. So we picked a task, a working memory task, that's thought to be a very strong driver of prefrontal cortex function and activity. And so this is what's called a NBAC working memory task. It's called a NBAC because the idea is for the subject to detect or indicate when a stimulus occurs NBAC before the present stimulus. I know that sounds a little confusing, so let me just walk you through this. Let's say we wanted to do an NBAC task. One back task. Task in that case is to detect when there are two stimuli, the same stimuli occurring in sequence, one after the other, or one back from the current stimulus. So if this were one back task, it would be, well, no, 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 okay, because there's nothing in sequence. But we could also use the same type of stimuli to do a slightly harder task, what is termed a two-back task. So in this case, instead of referring back to one stimuli previously, you refer back two stimuli previously. So you look back. Well, in this case, there's nothing to look back. This also is no. But if you look at the third stimulus, it matches two prior. So this would be a match. Here would be a match. Here, no. So I hope you get the idea. It's a fairly difficult task, actually. Yes, sorry, I forgot to mention that. One at a time. That's right. That's right. So in our pilot study, just with two subjects, we had them go through two scans each. One was at rest, and one was doing that task I just showed you, where they're lying in the scanner, the stimuli projected, and they can see through a set of mirrors these stimuli, and they respond with a button box while the scan is occurring in an MRI machine. So it's pretty much the same MRI machines that you may have gone into to look at various tissues in your body. The blue is healthy control subjects, and red is individuals with schizophrenia. So again, two subjects for each group. The hatched blue is baseline, solid blue is task. Same here, hatched is baseline, solid blue is task, and you see in these two subjects you have a fairly robust increase in GABA measured when compared to baseline. And in this case, here with these two patients, this one subject showed a very modest bump compared to baseline, and this subject showed, in fact, a decrease. I'm not sure if this is an actual decrease, but for this purposes, the most important factor is that it's certainly not an increase. And so based on this very promising set of results, and also this is probably among the first attempts at this approach for measuring GABA in schizophrenia, we were successfully able to get an NIH grant to study a larger number of subjects to test whether we can measure task-evoked GABA deficits in schizophrenia. And so briefly, these are the inclusion-exclusion criteria. Between the age of 18 and 55, diagnosis of schizophrenia with schizoaffective disorder, and no major comorbidities. And the other factors considered is being able to attend 2 or 3 sessions, testing sessions at Stanford or Palo Alto VA, and being able to stay still in the scanner for about an hour to 2. So the lower limit is very practical. I conduct part of my studies at the VA, and simply they just have an age limit. I don't know if you're aware of that, but if you want to study or have people officially involved in your work under the age of 18, you actually literally have to get an approval from the central office. They're very strict about that. The older age limit is due to the fact that it's pretty clear that GABA levels decline with age. So we wanted to mitigate that potential confound. Yep. I mean, that's a really important point. It may, and it's just one of those things that one of the many challenges and potential sources of variance that we can't control for, unfortunately. So it's presumably very possible that, let's say, if you measure the auditory cortex and someone is hallucinating, or maybe even singing a song to oneself, that you're engaging those systems, right? That maybe moving the GABA levels a bit. But that's only just a hypothesis at this point. Not directly. Again, that's one of the reasons why the GABA hypothesis is so appealing, because it's a different class of neurochemicals and different class of neural elements, receptors, and so forth that could be modified. Well, so we don't know. It's possible that, and these types of questions are very fundamental ultimately, and very difficult to tease out. Because yes, GABA may be downstream from some other, more fundamental abnormalities, but that's definitely possible. But we sort of don't know until we get a handle on this or some other specific neural process. Yeah, that's the idea. Again, right now we don't have sort of any sort of hooks, or very few hooks, and so we're trying to get more hooks into this complex entity, sort of as a starting point. So here I just wrote out our procedures, and so if you or anyone may be interested, please contact me. I've also brought some flyers for anyone who's interested in taking it, and I have a wonderful set of RAs working at this, and there's 3 of them. And then in terms of what instruments we use. So this is the MRI scanner. Again, this looks probably very similar or identical to most other MRIs. This is a subject bed for gurney, which slides into the bore of the scanner. And not shown here is that we have head coils mounted with mirrors that allows the projection of stimuli that I showed you. And it's housed in Jordan Hall, the psychology department. And this is the sort of the general area scanning environment. So let me then turn to the second experiment, second domain of research. Of which study? Oh, so this is ongoing. Yes. So in terms of sample size, I want to study as many people as I can and have funds for. So, I mean, but anything, target is hopefully somewhere between 20 and 30 per each group. So in relative to other studies in this area, it's a moderate sample size. It's not huge, but it's not small study either. So the second area I wanted to talk with is, again, a completely different area, a completely different perspective, different systems and processes. And it's involving it's a neurodevelopmental model or neurodevelopmental theory based hypothesis of the synaptic pruning, or some people refer to it as the Feinberg hypothesis. And it's sometimes referred to as the Feinberg hypothesis because Erwin Feinberg, a psychiatrist pictured here, first formulated this hypothesis around over 30 years ago now. And I was very fortunate enough to be mentored by him when I was at UC Davis. And if anyone has any interest in free time, please look up this paper he published in 1986. It's really such an elegant paper. The way he writes and thinks about these problems is sort of it's unfortunately a lost art. And so please look this up if you want an example of really elegant scientific writing in this area. Erwin Feinberg. Yes. Yeah. Hi. So what's partly remarkable about this hypothesis is that he formulated it in 1986 with in the absence of these fancy MRI machines. Back then it was pretty much only CTs, I believe. And back then, when I went to college, people were still referring to schizophrenia as a functional illness, meaning that it may not be caused by any specific neurobiological abnormalities. I was actually even taught about the schizophrenic mother. I couldn't believe that. In retrospect, that's what I was being taught. Yes. Well, so he formulated this very elegant theory based on very, very little empirical neurobiological evidence, but relied very heavily on logic, deductive logic, synthesizing, incorporating the best available clinical evidence to formulate this model. And so through the test of time, this theory has persisted, and now I would say, in my estimation, forms the basis of probably the best, most exciting theory in the mechanisms leading to schizophrenia. So let me tell you about some of the emerging work that has validated this theory. So some of you may be familiar with this New York Times article. It came out about 3-4 years ago, I believe, maybe 3 years ago, announcing, big new discovery. And let me take you through this a bit. It involved the identification of a specific gene that's thought to be one of the most significant single contributor to the generation of schizophrenia. It's called the C4 gene. It's a complement gene. Complement, some of you may realize, actually used to be thought as something not involved in brain processes at all. Complement is part of the inflammation process in the blood. But more recently, through work through Beth Stevens and others, one of the major breakthroughs in the basic sciences has been the realization that immunological processes, such as complement and related systems, are very alive and relevant for brain processes. It's thought that the complement in the bloodstream is responsible for tagging pathologic elements or elements that need to be cleared out by the immune cells. So it turns out that complement and the neuroimmune system employs very similar or the same mechanisms to clear out stuff, things that need to be cleared up or cleaned up in the brain, including unnecessary neural processes or neural elements, including synapses. So some of you may have learned that one of the remarkable things about the human brain is the plasticity and changes that it undergoes through development. At an early age, we are born with a full complement or near full complement of the number of neurons, but these neurons are very mature in that they lack cellular processes that allows neurons to communicate with each other. And so the number of synapses is fairly reduced when we're born. But then early on in childhood, the number of synapses proliferate greatly, many, many-fold. And then it peaks somewhere at age 5, 6, or 7. And then after that peak, it undergoes tremendous amount of pruning or regression in the number of synapses. And it's thought to, of course, yes, in a normal state. And it's thought to be a critical core component of learning. So we have a lot of synapses in the beginning, and then when we learn, it engages certain circuits and synapses, and the ones that are unnecessary or unused are pruned away. So let's see. And I have an illustration illustrating this point. It's thought that various complement molecules, including C4, are involved in this pruning process. Now, the discovery noted in the newspaper is that not only there's evidence of C4 involvement, but when they manipulated C4 in animal systems, they showed evidence of increased pruning in mice. So again, this is still fairly circumstantial, but what was really noteworthy was the sort of the vertical integration, we sometimes call it, where from a very specific gene to molecule to cellular processes and its association through the genetic association studies with schizophrenia. So it was sort of like an aha moment for the field, or one of the first or rare times thinking, wow, we're starting to get a fairly comprehensive picture, at least the rough outlines of a coherent pathophysiologic process centered on this gene and neural pruning, synaptic pruning. Yes, it can be. So I want to be careful to say in schizophrenia that has not been shown. There's no evidence, no direct evidence of that. But certainly that the involvement of these systems brings that possibility into play. And that maybe there is some dysregulation of this neuroinflammatory response that can trigger this pathologic cascade. Again, but that's very speculative. For now, there's very good evidence now that there may be involvement of the pruning process. I think they do, but I'm probably not qualified to really comment in detail about that. So let me take a little back step, though. So what are the evidence that supports the over-pruning hypothesis of schizophrenia? This is another sort of a seminal study, now about 20 years old now. This is a post-mortem study, like the GAP study I showed you before. This is a HC, or healthy control. And this study, you inject these neurons with a certain stain that allows you to look at cellular processes. In this case, they wanted to be able to count up these, what we call spines, these little protrusions right here. This is the site of synapse. So if you count up these little processes, you can get an indication of the number of synapses that are present in this person's brain. So this is a healthy control with a certain level of density of these processes. And these are two examples, one subject here, another subject here, using the same procedure, and you see a fairly drastic reduction in density of these synaptic relevant processes. These are some figures illustrating the points I made earlier. Here at birth, sort of lack of the density of cellular processes. Age 6 sort of looks like a dense forest. And at 14, you see diminution of this, corresponding to pruning that has occurred. So this is probably the major empirical finding that informed Dr. Feinberg's hypothesis. This is a post-mortem study where this scientist, Puttenlocher, in 1979, counted up synapses of various brain samples of individuals who passed away at these ages, from near birth to 80 or 90. And you can see this profile that I mentioned to you earlier, peak in synaptic density around 5 or 6, rapid decline, presumably in adolescence, because actually there was significant undersampling in adolescent years, but by the teens and early 20s, there was a decrease, so you can presume that this line probably exists. And then also that there's ongoing pruning or loss of synapse later on in life. And so this figure illustrates the time profile, the neurodevelopmental aspects of schizophrenia and other conditions. Yeah, so there are going to be different types of synapses. So with any combination, number of combinations of different types of cells. I think the synapses of primary interest are excitatory, glutamatergic, simply by the fact that they're the most numerous. In the brain, something like 70 to 80% of neurons are glutamatergic or excitatory. So they're, at first pass, will be the main focus. But it's also possible that it may be cell type specific. Yeah, so one thing I would advise is, I think it's difficult, if not impossible, and probably, I would advise against trying to put all these different theories and ideas into one model. Each hypothesis is more or less developed independently of others. And so it's very possible that, the majority of these ideas are false. And so we're still, unfortunately, at the very, very beginning stages of our understanding. And so again, it may be related, dopamine may be related to some of these abnormalities I'm talking about, but it may not be. So I'd caution you against trying to make too much sense out of all this. I'm sort of running out of time, so let me just skip through this and try to get to the end. And so, given the importance of this process and this hypothesis, it would be very important and interesting to be able to index or measure synaptic density in humans. And up until very recently, we have lacked the ability to actually do that. We've only had very distal proxy markers of synaptic density. But a few years ago, a group at Yale synthesized the first in-human, in vivo, non-invasive means or tracer that allows us to measure non-invasively synaptic density in humans. So this PET tracer targets what's called SV2A, synaptic vesicle 2A. The most relevant thing is that it's a molecule that exists in all or most synapses, presynaptic synapses in the human brain. And so these are just some examples of the results of this first published study in 2016, showing that it has all the qualities of being a very good tracer for this purpose. And so having been exposed to this synaptic pruning hypothesis and then having been introduced or notified of the availability of this new tracer to measure synaptic density, I was very interested in starting a new project to do just that in individuals with schizophrenia. So we've launched the Stanford PET study using this tracer. And I'm happy to report that after about a year of attempts, we've been able to synthesize this tracer at Stanford. For technical reasons, it's a very short-lived tracer, and so you have to synthesize it locally. You can't just purchase from Yale, and so it's sort of a big deal and complicated process to get the ability to synthesize it locally. So I'm happy to report that we were first able to synthesize it and finally get FDA approval to use this ligand just a month ago. And so we're just about to finally launch and study our first subjects using this tracer at Stanford. So we planned 2 experiments listed here. The first is a cross-sectional study. Cross-sectional meaning that we're going to measure or test individuals one point in time. One group would be healthy controls, and the other set would be individuals with schizophrenia. And then do a simple comparison. If the pruning hypothesis is true, then we should see reduced density in patients compared to healthy controls. So this is sort of the very easy, not easy, but just the low-hanging fruit that I and other select number of other groups in the country and the world are probably pursuing right now. The second experiment is a longitudinal experiment, and this is the one that I'm mostly excited about. It's a longitudinal experiment because we're going to measure synaptic density in patients over time. First at baseline, and we're going to try to prioritize scanning individuals with recent onset. And then a certain time later, approximately 12 months later, to see if there's ongoing excessive pruning in these individuals. And the reason this can be exciting is that the results of this study will lay the foundations for new therapeutics. And what I mean by that is the following. This whole area about neuroimmune-mediated pruning is actually a really, really hot topic right now in neuropsychiatry. Most active in the area of Alzheimer's, because it turns out, or the idea is that one of the pathologic manifestations of Alzheimer's is over excessive pruning. So because of that, a lot of companies have been very busy identifying targetable elements in this cascade for therapeutics to minimize or reverse synaptic loss. And so because of that, there's a number of agents that I've outlined here. I'm not going to go into details, targeting various aspects of this cascade that are in either phase 2, and I think 1 or 2 are even in phase 3 clinical trials in the area of Alzheimer's. And so the idea is that if you can show ongoing loss in the longitudinal experiments in schizophrenia, then it won't be too much of a stretch, again, as long as these agents are proven safe in Alzheimer's, to propose studying in schizophrenia. Again, I want to be very cautious here because I think our field is littered with false leads, false promises, and so forth. And it's very possible that this is not going to lead anywhere. But nonetheless, at least theoretically, I think there's an outline or path for rational, mechanism-based therapeutics in schizophrenia. So, and lastly, I just wanted to acknowledge some of the organizations and institutions helping fund this work, the SPARK program at Stanford, and IMH through its grants. Weston Havens Foundation is another foundation that's helping support the pilot studies of the synaptic pruning project. And finally, my home department. I appreciate your attention. I'm happy to answer any additional questions that you may have. Yeah, sure.
Just come up and talk to me individually. Thank you so much.
Sure.
Thank you very much, Dr. Yoon.